Anyhow, we're going to be giving a two-part talk. Both Manuela and I are looking into developing some new aspects of how we can follow up with irradiation experiments and investigate the cells. What you've been seeing so far today, from what I've observed, are some techniques for looking at endpoints, the gamma H2X, the worms, the embryo. And what we're going to do is talk about ways that you can look at responses in real time. These other techniques involved some processing and looking afterwards what happened. But what are some of these quick reactions, and how do you catch them? So we're going to be talking about a new technology, which is simply a glass probe tip, which is very small, on the order of the size of the cell compartments and components. I'll talk a little bit about the technology, and then Manuela will follow up with some of the actual plans for what we would do with them. Now, as I talk about this alternative sensing method, I would just encourage you all to be thinking about how you might use such technology in experiments that you would do. Again, this is an interaction that we have as a lab with outside users. We are a user facility, so we do a lot of collaboration and develop things as we go with others. At the outline of my talk is essentially to talk about some of the techniques that we have in collaboration here. There's going to be an ion selective probe, collaboration with a group that was formerly at Woods Hole in Massachusetts, Cape Cod. I'm going to talk about a positioner that Gerhard's developed. We're going to talk about another collaboration and yet another collaboration. And then at the end I want to talk about an environment issue, on how to keep the cells happy through all of this. So to get started, here's an image of several images of a probe. And basically with this slide I want you to get an appreciation of their size. Here you can see that this is a 1 millimeter scale. Here's a 1 micron scale. So the tip there is very small. In fact, you might even wonder, can light get through that hole? Okay. So let's carry on. How do you make these probes? These are essentially taken from capillaries, and you pull the capillaries using a special machine. This seems to be the standard. It's a Sutter Puller, a P2000. And by carefully extracting or pulling the ends of the capillary apart from each other in a very controlled scenario, you can obtain these very small tips. And that's the objective to start with, is that you need to have some kind of substrate on which you would then make a more refined probe. So after you've made the basic tip like that, the group in Woods Hole refined it further, in this case to design a probe to detect nitric oxide. And so their interest was to look at nitric oxide flow in and out of cell environments. So imagine irradiating a cell, and that cell emits a signal. And if that signal is nitric oxide, in this case, you'd be able to pick it up with this particular probe. So again, after you form the glass capillary, then there's more to do. There's more work to really obtain the final product. On our end, we have to figure out how to use these things. How do we hold them and actually get them close to the cell? Here's an image of our end station. This is in Microbeam 2. And here you see the probe coming into the side of a cell dish. And this is a water immersion objective. It's convenient to do all these probe works in an immersion scenario. So, for instance, when you're talking about signaling, you need this signals to be traveling through something. So you need medium in there. But we found out that it was a bit tricky to get this probe aligned just well with where the cells are, without poking through the bottom of the dish and damaging the window and causing a vacuum excursion, break, or also being able to manipulate that probe properly. It has to come in at a certain angle. And so to help with that, Gerhard devised what's called an offset hinge. to hold these probes and then to position them carefully. You may have actually seen this. It's attached to our end station, and it's sticking off to the side. And this offset hinge essentially is a way to pivot this whole structure, this whole arm, at this point here. But the mechanism is off to the side over here, the actual hinge and components. But the pivot point is down here in the center, right where your interest. This offset hinge is then mounted to an XYZ manipulator, so you can drive this into place, and then you could also go and visit one cell to another. So it has some versatility to it. Other uses of this hinge, once we got that in place, it could be used for some micro-injection facilities, for single-cell harvesting. It could be holding a fiber for some Raman spectroscopy work. And as Brian mentioned earlier, it could be used for rolling the embryo into place. And indeed, we have that capacity now to hold the top of the cover glass with a finger, which is mounted to that. And the finger moves the cover glass around, and beneath it well, your animation was much better than what I can do. Antonellis Man animation. Actually, I think Guy made that, but anyhow. This offset hinge has more versatility than simply holding a single probe. Now, another collaboration, this is Tuan Vo Din, and his interest is to look at light signaling from regions very close to the probe. So in this case, if you want to map a cell or a cell nucleus, you need to be able to move the probe around and excite only certain areas, meaning the regions just right around the probe tip. And oops. In his case, here's his tip. But what he does is he coats the end of the tip with some antibodies, and then he's able to bring the tip in close to a cell and actually illuminate regions of that cell that is really just at the end of the tip. So the tip end is 400 nanometers. At this point, light cannot get through there in a normal fashion, meaning the light waves that we use are on the order of 10 times larger than that. So meaning the wavelength of light is typical work maybe 400 nanometers for some excitation. So that's 10 times larger than the size of the tip. So what he's employing is the actual evanescent wave. And this is, if you study electricity and dynamics at the graduate school level, you tend to learn about evanescence wave. But basically, it's at the end of the tip, it's that in a region just around here, light does reach out and can excite molecules in fluorochromes that are just near the end of the tip. So this is a technique that can selectively fluoresce components that are right where the tip is. Now he has to do a few more special things to refine his probe tips, and in this case you're seeing the coating, the outside coating of this capillary, so that the light doesn't get out through the wall. It has to be a light-tight system until the very tip where the evanescent wave would be emitted from. Okay. Some more pictures of these tips. This is before the metallic cover is deposited onto there, and then here is after. So the actual tip does get larger, and typically when we're depositing metal coatings, we may not notice much of a difference in size, but here you certainly do with these images. It's basically because we're really magnifying this device. And here is how it would be all put together. Essentially, for this work, we need to couple light, or in the form of from a laser, into the sensor itself. And we have done that. Here's an image that shows a laser coupling with a fiber optic that then couples right to the tip. So that's technology that's pretty well established, and we will be using that for other capacities as well. Another collaborator. These are slides from Kambiz Purazai, who is at Drexel University, and we're actually planning to go visit him in the next few weeks. But his group met Brian at a meeting last summer, and we found out that we have some common interests, where his group was devising a number of ways to work with these probes. We now have a system where we can irradiate single-cell nuclei, and they could then use their probes to look at responses in these cells. So it seems like a natural collaboration, and we have certainly been following up on that. In his case, he has several designs. I just want to mention a couple. This one is involving a Raman technique, and it's surface-enhanced Raman spectroscopy, SERS. And what you can see is that at the end of the probe here, there are particulates that have coated that are coated to the probe tip, and these are on the outside. So the probe is itself being used as a finger to place next to your sample, and as you follow through with Raman spectroscopy, you get an enhanced signal around this probe tip. So here is an image of what that might look like through the microscope. Here is the probe coming in, and you can just put it next to a cell, or perhaps go into the cell. And here are some Raman spectroscopy images coming from various placements along that probe tip. It may be a little hard to see, but here is a probe tip coming in, and it stops about there. And this cross mark that comes down here is in a different location than the cross mark that comes down here. This is really at the edge, the end of the tip, and this one is just slightly before the end. But you can see there are different Raman signals. This is indicative that there is a change in the chemical and molecular composition along the length of that probe. So this is a technique that we are very keen on working with him and developing the Raman aspect. This is a new thing for us. You've heard of these other imaging capacities that we have, but we haven't done anything like this before, where we're looking at chemical makeup of a particular cell environment. So we're excited about developing this. It's going to involve introducing a spectroscope into our system. So to talk about his last one, this term nano comes into play, and he's calling these carbon nanotube cellular endoscopes. And in this case, what they're doing is actually attaching carbon nanotubes to the end of these probe tips. So the probe tip already, as I mentioned, was around 40 microns at the tip. If that wasn't small enough, glue a carbon nanotube or a couple to the end of that. And these nanotubes are even smaller than what you had here. So what can you do with that? Here's an image of how they make it. Again, there's more to do once you design the glass capillary that's been pulled to a tip. That now they have to attach this carbon nanotube inside that. So they have a technique to do that. And here is a schematic and then some results on how that would actually look. But here you bring your nanotube at the end of a probe, and actually would stick into and penetrate the cell and nucleus very easily. And once you're there, with Raman spectroscopy, basically it's shining a laser, and then you're detecting wavelength shifts. So the emitted signal it could be shorter or longer than the wavelength that you're using to go in. Some of you may have some Raman experience. So it's a common technique, Raman spectroscopy, but now being applied to a miniaturized system. And this is something that we would like to also follow up with our collaborator. So now I want to just shift gears a little and talk about our plan for an environment control. And we like to come up with acronyms, and in this case I found that AMOEBA works well for this particular plan, which is an automated microscope observation environment for biological analyses. What that means is that we want to have a modular system, a grand plan to satisfy some of our user requests. But basically, our users want physiological conditions. If you're going to irradiate cells and you remove all the medium and the cells get cold, how real is that? What kind of implications would there be? So we're working hard to satisfy these requests to develop a system that would keep the cells happy. Initially, we've been working with an air-CO2 mixture that blows over the cells and keeps them moist, and that's been pretty satisfactory. I would say that a lot of papers have been published on these works. When you compare to controls, it has worked very well. However, there's a limited time involved. It does allow particles to come through and get accurate counts, but you can't leave the cells on there for too long or else they really do suffer. Another technique we've tried is a heater ring, and this is pretty good for maintaining temperature, but then the medium that this heater is sitting in starts to evaporate. And as it evaporates, water is leaving, and then the concentration of the medium changes. The cells aren't happy with that. They start changing in ways that you don't want. So to solve that, this system is a flow system. It's to flow the medium across the cells in a very slow and controlled fashion. The plan is for flexible, user-friendly modular design, which offers a list of things here. You could aspirate medium, replace it with a different one. You could collect the medium later. You could have multiple dispensers to change medium or add other chemicals. We could introduce sensors to monitor the pH, for instance. And also this is compatible with microfluidics that could be down the line. So just to give a better idea of what this would be, here is a flow diagram where I show several different reservoirs where the medium might be stored, and it has to be pumped and it has to be either heated or cooled. This is really be up to the user on how they want to design their experiment. But the medium is brought into the dish. At the same time, medium is being removed from the dish, and it could either go through a lab on a chip for analysis, it could be dispensed. But each of these sections are modules, and the modules could then be connected with a computer for control. So I set something up just to check this out. Would it work? And here's just a picture of some of the components that I've started to play with. Here's medium sitting on a hot plate. Here's a peristaltic pump to move the fluids through. I have a temperature monitor here, which is actually connected to a small temperature a sensor in this dish. And in this case, cells were observed for 2 hours, and they appeared to be okay. So we may need to continue experimenting with this to try adding other components, other modules, and of course different cell types. But it appears that this is going to be a viable solution for keeping cells happy in our system. And I'm happy to hear more people talk about wanting to use this, so I'm glad about that. Here is also just a zoom in of the cell dish. You can see here's one pipette bringing in water or medium, and here's another one to remove it. Here's the temperature sensor. And I just wanted to zoom in here. This copper cap, which is actually in place on the Microbeam now, is a heater that's below the dish to bring some local heating. And finally, you can be creative. So this is just one idea of what this might look like, but all these components could be operated from this master brain back here. And so we feel that the AMOEBA is a flexible solution and has potential use in labs, well, everywhere. So I think that's the last slide, and I haven't really talked about too much of what the biology applications will be for these probes. I want to save that for Manuela's talk. But if you have any questions on